around. And the earlier version was, the early edition was somewhere in February 2020, this year itself. Courtesy this pandemic, we are having this online session this time. And yes, as of me, I'm very excited about the webinar today, because not only we have a renowned faculty in the form of the speaker, but also we have a very renowned faculty in the form of a moderator with us. And of course, the speaker will be introduced by our moderator, but I will take this privilege, the proud privilege, to introduce my own guru and Dr. Sukesh Nair, who's the moderator for the session today. Allow me to share my screen. So, okay. So Dr. Sukesh Nair is a name which is something which doesn't need an introduction, at least in the field of hematology, hemostasis, and laboratory around the world as such. But for the people who are from the industry side, particularly pharma side, just for the sake of formal introduction, he is on the working group of subcommittee of ISTH on APTT clot wave form analysis, particularly around factor 8 and factor 9. And he has had immense number of hands-on workshops for ISTH as well as World Hemophilia Federation. I am one who's benefited a lot from one of those workshops, and he runs proficiency testing programs in India. And the same program he has also helped in running in Philippines, China, Thailand, Sri Lanka, Egypt, and South Africa. Most of his publications, scores of them, they are on hemostasis. As far as the context for today's webinar or scientific meet is concerned, that is around Chromogenix. So he uses the APCR kit, which is developed by Dr. Stephen, and he strongly recommends its use by the laboratories which are already doing molecular testing for factor 5 latent mutation. He has also been using the Quamatic factor 8 chromogenic for a long time now. His lab, that is the Christian Medical College Vellore lab, is one of the expert labs which is used to assign the values of international standards to both ISTH and WHO for all the coagulation factors, coagulation concentrate, as well as the recently launched ADAMTS13. He also uses the factor 9 chromogenic assay, which is developed by Dr. Stephen. Currently, he's serving as the professor at the Department of Transfusion Medicine and Immunohematology, CMC Vellore, Christian Medical College Vellore. And I think it's almost 3 decades that he has been associated with this. So it's a pleasure to welcome you, Sukesh sir, and over to you now, sir.
Thank you, Ajay, for that short introduction and sweet introduction, and also bringing in the context of my indirect relationship with Dr. Rosen. Interestingly, Dr. Rosen was born in 1947. So Dr. Rosen, you don't know the significance of that year to India and today's participants. So he completed his PhD in 1978 in respiratory chain proteins. But fortunately for hemostasis, he shifted to hemostasis the same year. It was with Kabi initially, but later on joined Chromogenix. I'm sure he must have worked a lot with Professor Bjorn Dahlback to develop the APCR resistance kit. So I actually use that kit, which is plain APTT based APCR resistance, because our lab also does the factor 5 latent mutation. It allows us the ability to pick up other risk factors independent of factor 5 latent mutation, which could be factor 5 related or other acquired APCR. So it's a strongly recommended kit. It's a very nice kit. It's gone through a lot of proficiency testing program and it's done quite well. But what is his major contribution in hemostasis is the factor 8 assay, which could be assayed by the chromogenic method. Previously when before chromogenic assays came, it was a majority of the labs were doing one stage assays, and a few labs, expert labs, were doing probably 8 or 13 to 15 in the world, one in Australia, a few in UK, not many in US, but quite a lot in Europe. We're doing the 2-stage assay, which was actually manufactured by only one manufacturer in UK. And it was quite a tedious one. You could create a similar assay in your own lab, but it was a tedious one. So I would say that I would thank him for developing a chromogenic assay, which was actually a replication of two-stage assay on a chromogenic method. And of course, the chromogenic method became an assay of choice for many of the manufacturers of factor concentrate. And that is what he'll be talking to us today about. Our hospital has got a very good center for treating bleeding disorders, including rare bleeding disorders. So whenever there is any new product that is in the market and a new assay is developed, I am under pressure to get that assay. So among the people who must have ordered factor 9 chromogenic assay from Rosix, that's the new company started, I may have been one of the earlier ones, because it was I couldn't go through, and he didn't have a distributor. I had to directly get it from Rosix. Sometimes you have to directly deal with his company, and it was very pleasant experience dealing with Rosix. So we have been, though there was a distributor for Hyphen Biomed who produced a factor 9A chromogenic assay, we were not enthusiastic about it. So we went back to Rosix, and we are sticking with Rosix. So welcome, Dr. Stefan Rosen. And the floor is yours for the talk.
Let me see here. Thank you so much, Dr. Nair. It was a very, very kind presentation. And it's such a great pleasure for me to be allowed to make this presentation to you today. And I thank Instrumentation Laboratory and Muramur for suggesting and inviting me to give this presentation. I will discuss a little on the background of chromogenic substrate, the principles of the factor 8 method, as well as applications, and also some of the problems and tools surrounding assay discrepancies. So actually, I was employed at Cabo Diagnostic in 1978, and my task was to just finish a factor 8 project. So I was employed at a 6-month temporary position, but it took about 5 years to launch the kit. So I stayed 22 years in Chromogenix, and now further on 20 years at Rosix. So all in all, 42 years in hemostasis, and I have to tell, it's still as exciting as when I started in 1978. I would now like to switch to the next slide. Let me see here. I would like to make it as a slideshow. I'm not sure how this should be done.
On the left orange bar, there is a switch. I mean, there is a toggle. Go to the left, orange left top.
Orange left.
Still further up, right side, little on the right side. Yeah, this one. Yeah, up.
Aha. Thank you so much. I'm not a technical expert. Anyhow, so these are my disclosures. And 2 different types, as you know, are mainly used for determination of activity because the 2-stage method, the clotting, is not used anymore. So these are the 1-stage and chromogenic substrate methods. The 1-stage methods in their current form are used since the 1960s, and I would say still dominating in clinical labs, but the chromogenic substrate method is used more and more in the industry and R&D. Some general aspects which I think is important to recognize: neither of the methods reflect physiological conditions, since there is no endothelial surface or platelet present in these methods. 1-stage clotting method use non-physiological contact activators, and the clotting times are much shorter than in vivo. Chromogenic method use high sample dilutions, which may result in as much as more than 100-fold lower concentration of the analyte than what is the case in vivo. One such method are actually quite complex biochemically. This is not always realized because the method is so simple, but they are very complex biochemically, with all events leading to a fibrin clot usually before 90 seconds after addition of calcium chloride, as compared to 2 to 10 minutes for TNA generation in chromogenic method. And it's also, clotting also takes several minutes in vivo. One such methods are interfered by pre-activation of factor VIII, more about that later. In chromogenic method, relatively high dilutions are used, which contributes to a high specificity and to low interference. And very important, factor VIII is a labile factor, and analytical care is always important, including attention to pre-analytical conditions. Now short on the background. The chromogenic substrates for hemostasis were first developed in Mölndal, Sweden. It was a spin-off from a project at the company Bofors Nobel Pharma in the late 60s and early 70s on development of the first thrombin inhibitor for therapeutic use. Actually, already at that time, this company was located in the same building where Rosix is located now and where Chromogenix was located earlier. So it's quite some tradition here. So Alfred Nobel founded the company Bofors to make the dynamite, and some of the incomes and profits were invested into pharmaceutical industry. The crucial background knowledge was identification of invariant amino acid residues preceding the thrombin cleavage sites in fibrinogen for release of fibrinopeptides A and B. These amino acids were Feval and Arg, and they were invariant in fibrinogen from several species. So this peptide and many structure variants with a high affinity for thrombin were tested, but no substance with a sufficient half-life or without side effects could be developed at that time. However, the idea was born. We have now an amino acid sequence with a high affinity for thrombin. Let us attach paranitroaniline and make a sensitive method for determination of thrombin activity in vitro. And this was the birth of the first chromogenic substrate S2160, launched 1973 by Bofors Nobel Pharma Peptide Research Division. And this is a photo on one of the advertisements in thrombosis and hemostasis back in 1973. You can see this was the Bofors company advertisement. Now, this slide shows the absorption spectra in water of the intake substrate and of the paranitroaniline, the free chromophore. The paranitroaniline has an absorbance maximum at 380, but we measure at 405 nanometers. The reason is that the intake substrate has an appreciable contribution of absorbance at 380 because only a minor part of the substrate is cleaved to generate sufficient amount of PNA. So therefore we measure at 405 to get a negligible influence from the intake substrate. Now, in vivo, generation and regulation of factor Xa and thrombin is a complex system with heterogeneous catalysis. It's very beautiful. And the catalysis occurs on the surface of activated platelets. The role of factor VIII is as a cofactor to 9A for the intrinsic generation of factor Xa. So, all chromogenic 8 methods measure generated Xa as an endpoint, and thus a sensitive and selective chromogenic Xa substrate is used. So how was the first Xa substrate designed and on which basis? In vivo, Xa cleaves prothrombin to generate thrombin. In the 1970s, the amino acid sequence preceding the two factor Xa cleavage sites in prothrombin was announced at the conference. And the sequence was identical for the two sites, Ile, Glu, Gly, Arg. Now, in the mid-70s, Bofors Nobel Pharma company moved from Mölndal, and the Peptide Research Division was acquired by a Swedish therapeutic company, Kabi, and renamed Kabi Diagnostica. And their R&D manager was also present at the same conference, and he rapidly informed the company on the cleavage site sequences, whereafter intense synthetic work was started. This resulted in S2222, and of course many other peptides, resulting also in a good patent, which was very important for Kabi Diagnostica. I want really to emphasize this slide that chromogenic substrates are selective, they're not completely specific. This table shows selectivity data expressed at delta mil A per minute in optimal reaction conditions. You can see 2222 is a very good 10A substrate, but it also cleaved to some extent by thrombin as well as by plasmin. 2238 is a wonderful thrombin substrate. It's also a good substrate for APC. And even better for APC is S2366, which also is a good thrombin substrate. The plasmin substrate S2251 is also cleaved to some extent by thrombin. So absolute specificity is a wishful dream, but the very high specificity can be achieved by the combined use of a selected substrate in a properly optimized chromogenic method. Now, you may wonder, what about the numbering? Well, it is so that there were 4 labs at peptide at the Bofors Nobel Company in Mölndal. Laboratory number 2 did the peptide synthesis, so therefore all the substrates start with number 2. The next 3-digit number is simply the number of the consecutive synthesis. So S2222 was the 222nd synthesis in laboratory number 2, and so on and so forth. Now, S2222 was used in the first commercial 8 kit, the core test kit, and it was launched in 1985. This substrate was later replaced by 2765, which actually is a D-arginine on the amino acid and amino terminal. And this substrate has a similar selectivity and close to 2-fold higher K-cut, a higher affinity for 10A, and a higher solubility than S2222. So this substrate 2765 is used in both the core test and the chromatic kits. Now, as you will understand, the chromogenic substrate assay can be used for determination of very many hemostasis components. All of the methods are centered upon the cleavage of a chromogenic substrate by an active enzyme. So you can determine, for example, antithrombin, heparin, protein C, but in our case, we will focus on factor VIII, which is a cofactor to 9A, and 9A is the activator. Factor VIII is synthesized as a single chain protein with a molecular weight 330 kilodalton and secreted as a heterodimer with 280 kilodalton. It circulates in complex with von Willebrand factor, and upon activation, the B domain is released and factor VIII dissociates from von Willebrand factor. Refactor is a truncated 8 molecule which lacks the B domain, and the current gene therapy factor VIII projects they utilize B domain deleted factor VIII named BDD factor VIII. Now, the same year as I was employed, 1978, there was a publication by Segatchan and Miller-Anderson on the chromogenic factor VIII method. But this didn't work well. It was definitely not suitable to make a robust assay. I should mention that I don't have a medical background, and I was actually I was a protein biochemist in my education, so I had good use of purification knowledge. And within 2 years, we had the preliminary method. Professor Wienerzer in Vienna, in a city called Linz in Austria, had some good ideas here. And also my at that time, my R&D boss, Peter Friberg, and this was my wonderful collaborator, Margaret Andersson, who was a terrific medtech in doing all the lab work. And in 1985, we published a paper on a multi-center clinical application of the core test kit. And in the meantime, there were also methods published by the Stago group, by the Baxter in collaboration with Professor Hemkritz's group in Maastricht, and also by the Immune company in Vienna. And the product they developed was later taken over by Technoclone. So this is a simplified scheme of blood coagulation, and it's believed that the coagulation starts by the extrinsic pathway through tissue factor 7A, which generates the first traces of thrombin, and thrombin then activates factor 5, 8, and 11. And then the powerful thrombin generation starts through the intrinsic generation of intrinsic pathway for generation of 10A. So how to determine a specific coagulation factor such as factor VIII? Well, all chromogenic methods focus on the place in the cascade where the factor A plays a role, which is as a cofactor to 9A for activating 10 to 10A. And this cofactor is very powerful. It amplifies the 9A activity about 2,000–3,000 fold. And importantly, there is no use of factor VIII deficient plasma. The generated 10A is then allowed to cleave a 10A substrate, and the rate of PNA formation is related to the factor VIII activity of the sample. Now, as Dr. Ney so nicely mentioned and explained, the chromogenic method is similarly designed as a two-stage clotting method, which is a first stage with 10A generation. And currently, there are several 8 kits on the market. We have the IL Chromogenix kits. Hemosil kit is analogous to the chromatic kit. The Siemens manufactures a factor VIII chromogenic, Hyphen makes a Biofin 8C. And we launched a factor VIII kit last year. Technochrome has a Technochrome 8C kit, and this year Precision Biologic launched their cryocheck chromogenic factor VIII kit. The next slide shows the content of reagents and the number of reagents responsible for the factor 10 activation. As you can see, the Quartz kit is the most complex one with 3 vials, whereas chromatic is the most simple one with only 1 reagent and no other vials. All the other ones use 2 vials. You can also see that all 3 kits here, they use bovine components throughout, bovine 9A and bovine 10. The Biofin kit uses human components on 9A and 10, and the other 3 use human 9A and bovine factor 10. So this is just a comparison on the sort of simplification with the chromatic kit versus the Quartz kit. Fewer additions, a short activation time, and less dilution of the sample, which results in a higher rate of PNA generation, which increases makes it easy to get proper ODs in the instrument. So the procedure with the chromatic is very easy. The diluted sample, the combined reagent with all the components necessary to generate factor 10A, and then the addition of the chromogenic substrate after 2 minutes activation when you use a normal range, and you get the release of PNA. I should mention that I call this reagent for controlled dynamite, taking into account the inheritance of Alfred Nobel. So all the components here are present for generation of 10A. Factor 8A is a cofactor, very strong cofactor, but you actually don't need anything else than these components to generate 10A. So in order to prevent this, even on a slower rate, this reagent is made at a low pH. So the proper assay pH is obtained after dilution, after addition of the sample, which raises the pH to an optimal pH for factor 10 activation. And then, of course, factor 8A is a tremendous accelerator. And now to how these methods are used and some examples. And very importantly, the bioassay principle, which is a foundation in potency assignment, it requests that the sampler fulfills the condition of a parallel line dose response versus a calibrator upon assay with at least 3 dilutions. The current WIS standards are plasma-derived, and potency assignment of plasma-derived factor VIII concentrates works usually quite well because they fulfill the sort of like versus like condition. And the results are also mostly in proper agreement between one-stage and chromogenic methods. I will show some examples here. This is the calibration of the 6th IAS for factor VIII plasma. The assigned potency from 52 SI series with the one-stage and chromogenic methods was 0.68. And you can see for all the 5 kits here that there is a good agreement with the assigned value. Most of the labs used chromatic and core-test kits, and importantly, there was no difference between use of bovan and human 9A and 10. I should mention that, as we found out, and certainly also other labs found out in the early 80s, that if you combine bovan 9A with human 10, you get a much lower factor 10 activation, and therefore no manufacturer uses this combination in their factor 8 kit. Then I showed results with the chromatic and core-test kits only from the two lots number 3 and 4 of the SSC plasma, assigned activity 0.80 and proper agreement with these two kits, and for the lot 4, 0.88, and again good agreement with the assigned value. Then to concentrates, the results from the calibration of the 7th IAS concentrate standard, using presenting results both versus the 6th IAS, which actually was a recombinant material, and versus the 5th IAS, which was plasma derived. And you can see that there is some difference for the one-stage method with 1159 versus 1043 for the two standards, whereas there was a closer agreement between the Chromogenix results 1126, 1085, and most of the labs use chromatic and the core-test kit also here. Calibration of the 8th IAS, assigned potency 9.4, and good results in agreement with the chromatic and core-test, as well as with the Siemens and Technichrome kits. And again, most labs use the chromatic and the core-test kits. Now, life is not always so beautiful, so there can be problems with a higher, for example, a higher one-stage versus chromogenic potency summits of plasma-derived factor VIII concentrates, and this has been shown in various publications. And there are a couple of important approaches or tools to study assay discrepancies. One can study factor VIII activation as 10A generation versus time, and also stability of factor VIIIa. Some examples here, just to show you that the very high sensitivity in the one-stage method towards pre-activation of factor VIII has been published in several cases. This is taken from our 1985 publication, where we measured factor VIII activity with the core-test kit and the one-stage method at various time points after activation by thrombin. And you can see that this tremendously high increase of activity with the one-stage method, whereas the core-test kit only recorded the true decay in factor VIIIa activity, because factor VIIIa has a limited half-life. This dot here is a rerun of a freshly thawed reference sample, for establishing again 100% activity. So some examples of these deviations. This is a very nice publication from the NIBS group, headed at that time by Trevor Barracliff. And what they did was to investigate the time for 10A generation in a system with purified 9A, factor 10, phospholipids, calcium chloride, and including the thrombin inhibitor hiruridine to knock out any traces of thrombin. And the factor VIII concentrates were added at a high final assay concentration of actually 1 unit per mil. And they subsample versus time and followed the determination of generated 10A by hydrolysis of S2765. Very high purity products and intermediate purity factor VIII products were tested. And you can see here that for most of the intermediate purity products, it was a long time before 10A was generated to a marked extent, with the one exception, whereas for the very high purity, the monoclonal antibody purified materials, as well as two recombinant materials, there was a much shorter lag time for factor 10A generation, which indicated some pre-activation or at least a faster rate of factor VIII activation. And they illustrated this very nicely in a slide which shows the percent increase, the percent difference between the 1 stage and 2 stage, and correlated that versus the time required to reach half the maximal 10A generation in their study. And you can see that the longer this time was, the less difference was there between the 1 and 2 stage assays. But with the short time to reach half of 10A generation maximal value, you had a significant difference, 35–40% higher with the 1 stage. So this was an interesting illustration. And another case, actually, the secondary standard, the concentrate standard Mega-2 used by manufacturers for potency assignment of concentrate. It was actually so that this concentrate has a high 30% higher potency assignment with the 1 stage versus Chromogenix substrates methods. And the explanation was found from immunoblot analysis and studies on activation kinetics, which indeed revealed that the Mega-2 contains factor VIII material, which to some extent had been modified during the manufacturing, resulting in faster activation and lower stability. And this was properly reflected in the Chromogenix method, which showed actually about 8.6 IU per vial, whereas his 7th IU standard, which was his same starting material, had with both method 11 IU per mil. So this was an interesting illustration of the effect on the pre-activation. Now, so this resulted in a deviation from the like versus like principle and caused an over-assignment by 1 stage methods. Another cause of concern with the 1 stage methods is that they're often shown a greater interlab variability. This is a compilation of data published, presented at SSC meetings from either in-field studies or calibration studies of international standards, and compiled by Dr. Mickelson, who was a brilliant researcher at Kabi. And you can see that in all cases but one here, the Chromogenix substrate method had a lower interlab CV than the 1 stage method. And much due to the sensitivity to pre-activation of factor VIII and to the lower interlab variability, the 2-stage Chromogenix 8 method was recommended by the SSC in 1993 and adopted as a European reference method in 1994 for assignment of factor VIII potency in factor VIII concentrates. Some important features here. Requests that there should be made pre-dilution to 1 IU per mil with factor VIII deficient plasma with normal for available factor content. Final dilution should be made in buffer with 1% albumin, bovine or human serum albumin. And I should really emphasize here that the quality of albumin is crucial to allow it as a bulking agent in these in reagents. Selection of an activation time should be made to ensure maximal rate of 10A generation. And in USA, which to my mind is a bit surprising, both the USP and FDA still requires a 1-stage clotting method, but both regulatory bodies may now be considering a switch to Chromogenix methodology, or at least to allow it in parallel, because of all the problems with the new generation products and gene therapy, factor VIII and factor IX. And another matter is that it was found often in these collaborative studies that they found higher interlaboratory CVs on assay or recombinant factor VIII versus plasma-derived factor VIII. So we were a bit curious on this and made a limited study. We wonder whether this could be a stability issue. So this was again an observational study, not published, but I think this is something which I can show here. We used 2 plasma-derived concentrates, number 1 and 3, and 3 recombinant materials, 2, 4, and 5. And we determined the factor VIII activity at various times after reconstitution. And you can see that after 60 minutes, all the 3 recombinant products had indeed lost more VIII activity than the plasma-derived materials. And this could indicate that there could be lower stability sometimes on tested or factor VIII products, and that could cause higher variability in results if the time to start the assay in the various labs could be different. Now, switching now to different stability of active effector rate on the potency assignment. REFACT is a very interesting case, the BDD effector rate. This product was initially potency assigned with the variant co-attest effector rate method, which used an activation time of 20 minutes as compared to 5 minutes in the KIT method. And I was much involved in this work, and we had a publication in thrombosis research in mid-80s, and we were very proud because this method made use of the fact that the effector 10A plateau is reached after about 10-15 minutes due to inactivation of effector 8A. And the level of the 10A plateau was related to sample 8 activity. So, and this variant method allowed great robustness against slight variations which may occur in temperature and activation time when you run large assay series, for example, full microplates, because maybe you don't have full check of that the activation time is equal in all samples, but there was a problem. Activator effector showed to be inactivated more slowly than the calibrator, resulting in more extensive 10A generation and about 20% overassignment of the potency. And I should mention that this method was introduced to the Carber company in the 80s, long before the reflector was made. So it was used for potency assignment of monoclonal antibody purified factor H, such as such as octanative M. But the same method was then used for reflector, but it was a mistake. So this was one reason why the pharmacopoeia method was amended to require an activation time rate which warranted the maximum rate of 10A generation rather than using a plateau level. Now, another interesting case which is not related to concentrates but to a point mutation in mild hemophilia, but I show it anyhow because it's a nice illustration. Our lab, as well as the Malmö coagulation lab, were participants in an oligocenter study run by Professor Oldenburg in Bonn in Germany. And I won't go into detail about the study, but there were a number of patient samples distributed and measured at the various sites. We used a co-attest kit with 5 minutes activation time, the high range, which we had extended to a bit lower values. We had verified that. Whereas the Malmö group used the low range method with 10 minutes activation. And you can see all in general, the samples had a good agreement between the labs. There were 10 samples, 6 of which carried the arginine tryptophan mutation with about 2-fold higher values obtained in our lab. And why was there a discrepancy? We were, of course, very curious, so we investigated the factor 8A and half-life. And this slide shows the blue trace is the 10 activation versus time with the calibrator. The diamond is a mild hemophilia A mutation threonine to alanine, which shows a good agreement between the two centers. And then there are 2 patients' plasmas from patients carrying this arginine tryptophan mutation. And you can see that for these 2 patient samples, the half-life of factor 8A is shorter, resulting in an earlier generation of the 10A plateau, and then a slow decay, which is probably due to progressive antithrombin inhibition of 10A. So using then 5 minutes or 10 minutes activation time, you can understand that there will be quite different assignment of potencies. And this was actually published as a poster in Amsterdam 2013. Now, the case 3 is from gene therapy, where I had the pleasure to be involved in a project with Biomarine on the AAV5 BDD factor 8. And in 2017, there was a publication on the one-stage versus chromogenix acid discrepancy on analysis of plasma for patients receiving this therapy. And you can see from this figure in the publication that there's over the whole measuring range, there was a consistent about 1.65-fold higher value assignment with the one-stage method versus the Chromogenix method. And this was also obtained with a very similar result on analysis of plasma patients receiving the Sangamo Pfizer AAV6 BDD factor 8. And it was quite confusing because mostly one-stage results are known to be lower than Chromogenix results on analysis of effective Oxynta. So why were the one-stage values about 1.6-fold higher than the Chromogenix results? Well, I decided to use a prothrombinase complex for an investigation on the very early phase of ternary generation in gene therapy patient samples from the biomarine case. And this prothrombin complex, as you know, there were factor 10A is the enzyme to cleave prothrombin and generate thrombin in the presence of 5A, calcium, and phospholipids. And the factor 5A cofactor activity raises the 10A activity more than 1,000-fold. And this method allows determination of picomolar 10A concentrations. And the method is designed such that 10A is rate limiting for thrombin formation. And the generated thrombin then cleaves a thrombin substrate to make release of PNA, and the rate of PNA release is connected to the 10A activity. So the quartz SP factor 8 kit and the one-stage method were used for investigation of 2 patient samples. In both cases, a wild-type factor 8 was used as a comparator, but suitably mixed with factor 8 deficient plasma to give the same factor 8 activity in the quartz kit when used in the prescribed 5 minutes activation time. So the 2 comparators in the 2 patient plasma cases served as internal standards in the same SR series. Now, the result then on ternary generation in a one-stage method with actin FSL, you can see that there was a faster ternary generation in the early phase for the study samples versus the comparators, even though close to identical Chromogenix activity was obtained with the co-test SP factor 8 method using the 5 minutes activation time. And this results on early ternary generation, of course, were reflected also in early thrombin generation. So the early onset of 10 activation and thrombin formation illustrates here a deviation from the like versus like principle, which in this case, due to the faster thrombin generation, causes an overestimation in the one-stage method. And this study was recently published in Blood Online in September, and it will appear in as a printed version, I think, at the end of this month. And there will actually also be podcast, as I've been informed, on this work. So it was good fun to do this study. And very short on the potency assignment of extended half-life recombinant 8 and 9. And this is really constitute challenges of the bioassay principle with many deviations from like versus like. We have been involved in some studies here. It's been shown very clearly large one-stage versus Chromogenix acid discrepancy, as well as discrepancies between one-stage method. For example, in EHLR factor 9, the pegylated factor 9 called NNGP and the factor 9 fused with albumin. There we could show, and other groups show that the variation is due to discrepant interactions of product and the calibrated with the content activators. I just show one example on the extended half-life recombinant factor 8. This is a slide very kindly provided by Dr. Elaine Gray. She arranged in a clever way, as usual, a big multi-center study on the extended half-life factor 8 using several one-stage method and several Chromogenix kits for analysis. And these are specifically the results from their lab on one of the coded samples named called ME. And you can see that all the 4 Chromogenix kits showed a good agreement. The target potency was 10. And there was a large variability between the one-stage methods. And for 2 APTT reagents, APTTSP and PTT, there was almost no value assigned when assayed against the 6 IS for factor 8 plasma. So these new generation products can be safely measured with Chromogenix kits, but one has to be careful when using the one-stage methods. So I come now to the conclusions. All chromogenic A kits use 9A as enzyme and 10A formation as endpoint, and with the factor VIII containing sample as rate limiting, and generally lower CVs is obtained. Human-human, bovine-bovine, and human-bovine combination of 9A and 10 are used, and all combinations appear suitable for accurate factor VIII concentrate potency assignment. A factor VIII concentrate should be pre-diluted in 8 efficient plasma to 1 unit per mil. The sample diluent buffer should contain 1% bovine and human albumin, and again I emphasize the quality of the albumin. The chosen factor 10 activation time should provide the maximal rate of factor 10 activation, and this is usually when about 40 to 60% of maximal 10A has been formed. Chromogenic potency assignment of plasma-derived concentrate against a plasma-derived calibrator works well, since there is a like versus like condition, and the condition of a parallel dose response of sample versus a calibrator is usually met. This is also the case for chromogenic potency assignment of recombinant concentrate versus a plasma-derived calibrator. Investigation of 10A generation and of factor VIII stability are very helpful approaches and tools to ensure that accurate potency assignments are made, and to really see that the like versus like conditions are prevailing. Discrepancies often occur on analysis of extended half-life factor VIII, factor IX, as well as gene therapy express factor VIII as well as factor IX. Such discrepancies appear to be due to different interactions of sample and calibrator with the content activators, and to differences in very early 10A generation. And last but not least, it always has to be recognized that factor VIII is a label analyte, and analytical care and strict adherence to validated procedure is crucial. Now, before I finish, I was just some information on Rosix. The company started in May 2000, so we celebrated 20-year jubilee now, and the background is my work at Carbon Diagnostic and Chromogenix. We focus on hemostasis kits and reagents, not surprisingly, and these are our chromogenic kits. Should mention that the prothrombin kit is actually designed upon the prothrombinase complex, so only biologically active prothrombin is determined, in contrast to the hecarim-based activity methods of prothrombin, which also activates even acarboxyl prothrombin. And it should mention that the factor 9A kit and the 11A kits are used by manufacturers for detection of trace amounts of active factor 9 and for presence of 11A in immunoglobulin batches. And especially in the cases of immunoglobulin investigations, the pharmacopoeia methods, the NAPT method, is simply not sensitive enough, which has caused tragical consequences in 2010. We do some reagent manufacturing. We, for example, make also phospholipid for the NAPT and for the TDT. We make some buffers and validated 20% bovine serum albumin and also some proteins. We also make some customized products, and the core activities, of course, method development, and we make contract analysis. The biomarine was one case, and similar to Dr. Nai's lab, we are also one of the expert labs in calibration of WHO standards. So with this, I thank you so much for your attention, and you're welcome to make any questions. Thank you again.
Thank you, Dr. Rosen, for that elaborate talk. In fact, that is what we call science behind the science. So if there are any questions, please put in the question and answer box. Sir, over to you. Sukesh.
I think there's one question already there, Ajay.
Okay. So there's a question from Mallikarjuna. Please let us know the regulator's expectations on the use of chromogenic substrate assays as lot release assays. Please share us if there are any reference papers which are published on this.
Well, do you mean factor 8 or factor 9 or only factor 8?
Mallikarjuna, if you could just put in your answer whether it is factor 8 or both factor 8.
Probably factor 8. I would guess probably factor 8.
Okay. Well, as you know, in Europe, this is a pharmacopoeia method since 1994, and it's still not the case in USA. And being now a member of a voluntary expert panel in USP, there's some work ongoing to which I think will result in some modification and perhaps allow both the to use the chromogenic method in parallel with the one search method. And actually, the Hemophilia Society in USA is urging the FDA to approve chromogenic assays both for 8 and 9 for monitoring in the labs. So I think there will be some change over time here, but it takes a long time, obviously.
So there is another question. Is data analysis done using PLA or only by standard microplate reader? Which microplate reader is recommended?
PLA, can you explain PLA?
Is it platelet?
Yes, I would say that we use the CombiStats statistic program from EDQM. We subscribe to that. Of course, we also use the software in the microplate instrument, but for release testing and for when we participate in the multi-center studies, we deliver the results based on the CombiStats statistic program.
Okay.
That's a kinetic assay program.
Pardon, that is the?
It's a kinetic assay program.
Well, it is. We use a parallel line actually. We don't use. We can use a slope ratio as you indicate, but mostly we use a parallel line, and because that is almost always what the NIBS statistics are using when they ascribe the potencies. So that's why we do this.
There is another question for estimation of factor 8 in a foamy viral gene therapy trial. Please suggest the best and the cost-effective method in vitro and in vivo.
Well, if we relate to patient samples, I would say that the most cost-effective way, but not the best way, would be to use only one dilution, one sample dilution only, which is, I would say, unfortunately, what most or what many coagulants do today. But this is not the best approach, and I support Dr. Kitchen in Sheffield, who eagerly asks people in the coag lab to use at least 2 dilutions to really check the accuracy of the assay and whether there's a consistency versus the dilutions of the calibrator. So I would recommend that at least 2 dilutions, and actually preferably 3 dilutions should be used. This then is not the most cost-effective way, but it's the best way.
This question is for both, I think Dr. Stephen as well as Dr. Nair. With your experience and in your opinion, which country do you think is going to be the next hub for boom with regards to factor 8 research and usage? So both of you are more than welcome to answer this question.
Well, this is really difficult for me to comment on. I really have to, no, I couldn't comment. I mean, it's a very exciting area where we live with now, with a lot of progress being made. It's a lot of concern, especially for these new generation products. And I I think this may lead to increasing use of the chromogenic methods because it's so complicated in the clinical labs nowadays with all the different results obtained with the new generation products. But to pinpoint the future is really difficult.
Okay. So, Kesh sir, yeah.
Yeah, there's a lot of gene therapy projects that are happening in India. So I think India will be in the forefront of a lot of gene therapy work on hemophilia, and both A and B. So there will be a lot of effective use of these assays in our country too. Yes.
Okay. There's another, so this is not a, okay, any other farm, is there any pharma customer which has been using kit on automation platforms? So I'm not
yes.
sure. Yeah.
Oh, yes. This is what they do actually. So they use, some of them use robotized microplate instruments, which very high throughput. Other use, for example, the ACL instruments, and I know that for sure. And also some others, I think, use the BCS XP instrument from Siemens, maybe also the Star instruments, but for sure, analysis are run by automated instruments by pharma companies.
Can I make some comments?
So please.
So, Dr. Rosen, though Steve has made a phenomenal article on the fact that in his country, the cost of chromogenic assay and one-stage assay, there is hardly any difference. But it doesn't relate that to definitely India and most of the developing countries. So if you see the usage of chromogenic assay in India is probably restricted to my lab and probably one or two more labs. But the maximum benefit that we have benefited, other than satisfying a clinician who use factor concentrate, is the biggest contribution is to identify the discrepant mild hemophilia A, because the pressure on the lab is to identify the cause of bleeding in a bleeder, and in a person who has a discrepant mild hemophilia A, the chromogenic assay comes really handy. So that is one issue. So the use of chromogenic assay in the developing world will take some time because the cost is an issue. I don't know what the manufacturers of chromogenic assay will be looking at when many of the modified extended half-life hemophilia, factor products come, or even modified hemostatic products come for patients with hemophilia. But like even thrombin generation test, tests like thromboelastogram, all those bypass effect assays are not frequently practiced now in our country and definitely in the developing world. So when you mentioned about the effective use of the article published by Trevor Barrowcliff, that is the like versus like principle, where he spiked factor deficient plasma with factor concentrate to get close to 100 international units per deciliter or 100 international units per ml. Would that be some way of overcoming the absence of chromogenic assay in our country, and maybe help the labs to slowly get into chromogenic assays, realizing that one-stage assays have difficulties, have limitations?
Yeah, it's well, it's a good point. I would say that the cost issue is a bit. It's so. Several decades, one has always thought that the chromogenic assays are more expensive than the one-stage method, but at least in Europe, this is not the case, as was shown in the publication by Dr. Kitchen. So, but this rumor lives on, and actually, of course, we use a lot of deficient plasma in our comparative studies, and it's really not cheap. So one should really take a closer look again from labs to really check when applying the chromogenic kits on an instrument, especially for factor 8. Factor 9, I have to tell, is still more expensive with the chromogenic, but factor 8 is really not so. You can miniaturize the assays on an automate with the factor 8, and you have good throughput. So I think I think this notion will change over time if the message from the Dr. Kitchen's paper will be spread thoroughly. And I completely agree with you that for diagnosis of mild hemophilia A, it's really important to use both One-Stage and Chromogenix. It has been so fruitful for exploring these cases. It's very, very.
Oh, yeah, my lab has heavily benefited from that particular product. But the fact is that chromogenic assay is not only expensive, not because the purchasing the kit is expensive, but the whole kit cannot be consumed the way it's consumed in Europe and in UK. So there is a wastage of a lot of. So it will take a long time. It will take a long time for chromogenic assays to come. Now, for example, I think somebody like Peter Turicek, who is with not Baxter now it's
Takeda.
called Takeda. Takeda. So he basically has kept in mind that the products that Takeda produces can also be monitored using one-stage assays. So when he's talking about one-stage assays, it is using the standard human plasma, or is it the plasma which is factor concentrate in factor deficient plasma?
I'm sure he must recommend use of a concentrate diluted with factor 8 deficient plasma. But then I would also say that for any concentrate, you can of course use a product-specific standard to then safely use a one-stage method, but it's not so practical for the hospital labs to use product-specific standards because it may question the value of the international standard, and also if the hospital uses several different types of concentrate for treatment of the patient, it can be a nightmare for the labs because they don't know where they get a sample and they don't know which was the product. So they're just told to measure factor 8, for example. I should also make a comment which you completely correctly say, that there can be a lot of waste in the Chromogenix 8 kits because that will make it expensive if you can use a whole kit. But actually, at least I think with the chromatic kit as well as the co-test kit, you can aliquot and freeze at -70, suitable amounts, super volumes, and just thaw to some slight excess what you think you will need for the next series of tests. Of course, you have to validate and show that it worked, but we're done so especially for the factor 9 kit, where there are even less samples in each SS series, and we actually have a procedure to give to users that aliquot and freeze and validate, and then you can use even a stored standard curve on the instrument if you do the same procedure every time, and then you can cut the cost considerably. And actually, I have an interesting example from Malmö. They use the co-test kit since 1980s, and in the 3 decades they have used. They are very, very careful. They order many kits on one batch. Then they run 6 complete standard curves after reconstitution, pooling all reagents. Then they may run 6 standard curves after freezing and aliquoting, and then they establish a standard curve on the instrument, and they use that stored standard curve for the whole year. And they just use a control plasma, of course, to validate each SS series. And believe me or not, I could witness the results when I visited them a number of years ago. Over 1 year, the CV between SACV of the control sample was 5.5%. Using a stored standard curve and frozen reagents thawed each time. It was fantastic results. So you can really cut the cost.
That's
Right.
good news.
I think
now free advice, free consultancy for you. You have a product which is Chromogenix prothrombin product. Have you ever tried them on hemotoxic snake venoms? because if you look at some of the hemotoxic snake venoms like Echis carinatus and Russell's viper, you could probably benefit a lot by using of these kits in hemotoxic snake venom.
I would agree on that, because, but admittedly, we have not pushed this kit very hard, but it's based on a very sound biological assay principle, because it uses only the prothrombin complex activity, and therefore you measure only biological active prothrombin. So it was an interesting idea to use the snake venom. Yeah, maybe we should follow up that. Yeah, very interesting.
Last comment I have, Ajay, it was very interesting that somewhere in the talk, Dr. Rosen used the word dynamite to describe the effectiveness of the chromogenic assay, and Alfred Nobel created the dynamite. And
Yes.
I realized that the company is now Bofors Nobel Company, and Bofors is a manufacturer of some of the best artillery equipment in the world. And India has won a few battles because of Bofors gun. So this seminar is, you're being born in 1947, and this seminar being having this background is very, very important.
So that is the art of stitching the dots together, and sir is a expert at that. So the next statement or question from Sanal is what perfectly goes with what you've been discussing, that there are problems that there are only few samples to test at a time. I don't know, Sanal MG is from a pharma background or from the laboratory background. And the gap between tests are in months. The problem is that the reagents of the current test kits are unstable. I want to understand what does that mean exactly. The problem with Western blot is that it gives multiple bands. Okay, Sanal, we would request you to elaborate your question or statement a bit. But before that, there's a question to both of you. All the scientists that I've spoken to who do serial dilutions for factor VIII assay have all found it very difficult to do the process, thereby getting inaccurate results. Any suggestions or automations or tips to ease the process? I'm sure you're talking about the anonymous attendee, you're talking about the demanding as regards, the maybe the operator efficiency or maybe the performance of the procedure as such. So any light on that, either of you? Dr. Stephen?
Well, maybe, well, I don't know whether this may relate to the exact conditions used in the lab, but in principle, it should be straightforward to make serial dilutions. If it relates to concentrates with a high activity, then the serial dilution should be made in factor VIII efficient plasma, and then to 1 unit per mil, and then followed by buffer dilution. If it relates to patient plasma, it should be okay with only making it in buffer, of course. But honestly, if everything is under good control, there should be no big problems with making serial dilutions, because this is an important procedure to check for parallel line versus the calibrator. So it's a bit difficult to give a good answer here.
Okay, sir, any comment or any suggestion from your side on the serial dilution part? How to make it more effective?
Serial dilutions is definitely useful for parallelism, because we can identify the effect of any. Thing is, the advantage of chromogenic assay is that many of the factors that interfere with a one-stage assay does not interfere with chromogenic assay. But activated sample may possibly show its effect if there is serial dilution done.
Okay. So, there is one last question for the day. Is chromogenic assay an alternative for clot lysis assay? Interesting.
Well, that's not the case, but actually we had developed an alternative to chromogenic alternative to clot lysis assay in the mid 80s and early 90s, when there was such strong interest in in the fibrinolysis area. But we actually, one important thing which we found and which was not generally accepted because of logistics, I think, is that when you take a plasma sample or to withdraw blood from a patient and you intend to measure fibrinolysis activity, most people don't realize that TPA is so rapidly inhibited by Pi1. So we could only get good results when using this special tube called Stabilite. I don't know whether it really exists anymore. And then we could develop a very, very good bioimmune assay for TPA, for example. But it requested that you had to make the blood sampling in a medium which prevented Pi1 inhibition ex vivo of TPA. So unfortunately, this was never made into general use.
Ms. Neetu, if you have a question, maybe you can ask it directly. You can unmute your CI.
So with regard to dilution, serial dilution that you had given us a suggestion, do you suggest any particular pipette or automated dispenser for the accuracy of dispensing the exact volume or exact serial dilution to result?
Okay, you see, we are not the best example here because we don't run so large series as you do in a large lab. So actually we do manual pipetting, but I'm sure that the automated dilution equipment, which are validated, is a very good tool for making all these dilutions. But we use actually high quality Eppendorf pipettes in our lab, in our manual work.
Okay.
Thank you. Thank you, sir.
So I see there are no more questions. Before I close this seminar for today, if there are any questions, still maybe you discover some question a little later, you can just write it to us. We will share the questions with the speaker as well as Sukesh sir, moderator for the day. And of course, either the presentation or the recording of the seminar today will be available in some form to you all who have registered and attended the session today. The form and when will depend on, I mean, we'll make it a little clear shortly. And so, Sukesh sir, any closing remarks from your side?
Nothing as such. It was an enlightening talk, going through the history of the development of chromogenic assays and how the industry was driving the need for such a development of such an assay. And I think if industry is driving it, obviously this is the assay to stay. But I think for our country, we need to develop methodologies on how to make the assay cost effective. Maybe manufacturers like IL, WERF and ROSX will need to talk to experts and see how this assay can be introduced. And see, the thing is, one thing I've realized, long time back in CMC, that's when I joined, we were using factor deficient plasma for factored assays harvested from patients. We don't buy anything. We don't make anything from in our own lab. We don't make pool normal plasma. We buy everything because we don't have time for pool normal plasma. We don't have time for harvesting. So the thing is, once a lab is providing good results, it will start getting samples. It will become very busy. And they will start only using products which are good. So I think it's a mandate and for manufacturers to send their representatives into countries which are not able to afford good assays to see how it can be affordable to them, like the methodology that Dr. Rosen had mentioned, and how they can help them into such methodologies so that once they start producing results, see, many of the work in such countries are also driven by industry. slowly the industry will start providing them a lot of patients. A lot of essays will happen, and then they'll forget about the cost-effective method modules or algorithms. They will straight away go for the regular algorithms. So I think the manufacturers have to realize that introducing cost-effective doesn't lose business for them, actually can gain more business for them.
So, Mr. Muthu, you want to say something? I see you're on mute.
No, nothing, sir. Thank you so much for everything, Dr. Sukesh sir, as well as Dr. Rosen sir, for this wonderful webinar. I hope industry people are much more benefited, especially for the people who are working on the Factory 8 assays. You have covered almost everything, even like what Dr. Ajay, you were saying it. Suppose if even future, suppose they have any doubts, let them send a mail, we can get it. Thank you so much, Dr. Sukesh sir, as well as Dr. Rosen. Very, very informative. Definitely the people who are working this get knowledgeable. That is our ultimate objective.
Thank you,
sir.
Thank you.
So once again, on behalf of Werfen India, Werfen Academy and Moore & Moore, I would like to thank you all for participating in the Chromogenix Scientific Meet, second edition, which is a Chromogenix webinar today. And thanks to Dr. Stephen and Dr. Sukesh Nair for really putting some inciting thoughts and inputs for us. I'm sure the industry and both the laboratory people will benefit out of it. So with that, this is the close of the webinar, and namaskar.
Thank you. Thank you.